Thank you, Melita, and to the organisers for inviting me today to give you a quick overview of the technical background for today's symposium. So I'm basically going to take you through what it actually means to edit the genome and how we can do this. And I'm going to sort of end with talking a bit about the limitations and also a little sort of view of future work. So I thought that might be quite useful for this group and the discussion we're going to have. So find and replace, what is gene editing and how does it work? So I thought I'd start just by discussing, obviously, very simply, in terms of, what are we actually trying to achieve here? And so obviously if one has a genetic illness, there is a gene pictured here. Have I got a pointer? I do indeed, which potentially has a deleterious mutation shown here in red. And there's obviously, ways one can replace or repair that gene to alleviate the genetic defect. And typically this is done with a so-called repair template, which is basically just a copy of the mutant gene. And one relies on a process called homologous recombination to effectively encourage the genome to recombine its chromosomal mutant copy of a particular gene with a kind of repair template, which contains a normal copy of this gene without any mutation. Now, that's relatively simple, but unfortunately, it's a very, very rare event.